Oh, hello and welcome everyone to our today's session of our varvis webinar series on liquid biopsy, pushing the boundaries, the limit of detection in NGS liquid biopsy. Today we invited three speakers, three experts that will talk about the latest advancement in the NGS liquid biopsy technologies and how these novel assays can be properly validated and tailored to the specific use. But before we start, let me shortly introduce to you who we are as we host this webinar today, and also who I am. My name is Nadine Neser. I'm Marketing Manager at Liesefeld Technologies, and I will guide you through the program today. Liesefeld Technologies is a software development company located in the north of Germany, right at the lovely Baltic Sea. We are a medical device manufacturer, which means that we develop and operate our products according to international standards. Our product varvis is a cloud-based clinical decision support system, a complete solution for all clinical NGS applications, from targeted NGS panels to whole exomes and also cancer diagnostics. It is a CE-labeled medical device specifically made to aid in the diagnosis of patients. So yeah, and the young team already a quite outdated picture with already a lot of new colleagues. So like I already said, we have three speakers and three talks today, beginning Dr. Andreas Polten with NGS solutions from Agilent for clinical applications. Our second speaker is Dr. Ben Liesefeld, reconciling regulation innovation with standardized software, and last but not least, Dr. Dennis Schanze, matching analytical performance and clinical utility. We will end the session with a Q&A panel, so we invite all our speakers to answer your questions. So if you have any questions, feel free to raise them whenever you want. You can do so depending on which device you use. When you're using a mobile device, nice, at the top with the question mark at the top of your application, then you can just enter your questions into the chat. Or if you're using the browser application on the right side in the question tab, there you can also enter all your questions. Okay, so I would say let's jump right in with our first talk and our first speaker. Welcome, Andreas.
Yeah, so, I'm sharing my webcam and say hello.
Hi, shortly introduce you to our audience. Andreas has a PhD in molecular biology from the University of Göttingen. He joined Agilent in 2000, so he's already part of Agilent since more than 20 years and has an extensive experience as an application specialist covering the genomics portfolio, including NGS-related products. And since 2017, he is part of the NGS clinical workflow team for oncology market solutions as a business development manager. And yeah, I will hand over to you now for your first presentation. Just let me briefly give you keyboard and mouse. So now you have access, and yeah, thank you.
Okay, thank you for this nice introduction, and thank you to all the attendees for attending this seminar. I think I don't have to say anything for myself because Nadine has already done this in a very nice way. So just have to got one question. How do I move the slides forward and backward? Does not work. Do you see one? Nadine, can you? Does not. I cannot move the slides.
You should have the mouse now. You can. I also see you moving the cursor.
I can move the cursor, but I cannot.
Click into the slides. You can't move it.
No.
No.
Okay, now it works. Maybe there's a delay. Okay. So when I prepared this talk, I used the chance, and sometimes it is very helpful to do this. Also, if you work a long time for a company, to look at, what are the, what are the contact points from within Agilent towards cancer, because it is definitely one of the big projects internally to have a focus into cancer and cancer-related applications. And these are some of the pillars within the company. And I definitely will not go into detail for the sake of time here, but it basically ranges from real-life cell analysis with products from companies which have been purchased a couple of years ago, from Seahorse or biotech, basically, which allows you or gives you the opportunity with technology to look into real-time cell analysis in live cells in culture. Another big pillar are the pathology products from our colleagues from formerly Darko, which are one of the, let's say, big players in the pathology market with H&E and special stage, IC robotics, digital pathology. Also, we do make use of our, let's say, of another branch, which in Agilent serves mostly pharma companies, which is focusing on GC, HPLC, and LC, MS, triple quad, and these kind of analysis for, sorry, no, it moves back, okay, for these kind of applications. And we also have one branch which is usually working somehow kind of in the background, which is companion diagnostics, which means that we do work with pharma companies in the process of developing drugs and kind of co-developing the appropriate tests. And of course, project-related things which are here for the biopharma solutions, which are dedicated projects where we help pharma companies to develop entire portfolios. And then last but not least, genomics. Genomics is also broader than what we are talking about today, but today we will focus on NGS. And interesting enough, can I move back? Sorry, I don't know, I have to move, sorry for that. Okay, so now it works a bit better. So if we look at the portfolio around NGS, so we do cover more or less the entire process from wet lab sequencing to software solutions, apart from sequencing. And sequencing, as you may know, we completely focus in these days on Illumina. We do look at other large vendors as well, but for the time being, it is Illumina we work with. When you look at the wet lab part, we basically segregate this into 4 portions. And if you follow the workflow, it starts with QC, library prep, and automation, and the corresponding panels. With QC, I think I will not, for the sake of time, dive into, but I kind of like to say we are the kind of Illumina of QC, because we do have the complete workflow starting from bioanalyzer, from the fragment analyzers and the tape stations, basically for every kind of analysis, both QC and quantitation for input material, but also for the individual steps within the workflow. The library prep, this is where I will dive a little bit more deeper into, because this is, I think, one big portion of this today's seminar. The kit which has been used, which has recently, not so recently, long now, it's a year or so ago that we have launched the XTHS2 library prep kit. This kit is in several aspects somehow really new and unique for us. One is that we do fully support both pre-capture and post-capture pooling. So it's really your choice what you prefer. We have added an enzymatic fragmentation kit, so you can do enzymatic fragmentation where we do see some benefit on FFPE material, or generally said also for low input material, but you can also use your existing methods to fragment the DNA. The big part here is that this is kind of our center stone for all of our applications. So it does serve the standard applications, DNA as input, target enrichment, and sequencing. You can actually also step out here pre-enrichment, so after the pre-capture PCR, and do whole genome analysis. Some people, for example, combine target enrichment and then taken also out, some extra material from the library prep and do low pass sequencing based on that for copy number analysis. But it also, is used at the library prep kit for the RNA applications, either including target enrichment or excluding target enrichment, meaning you can do straightforward whole transcriptome analysis, poly A, if you start from poly A material. The upper workflow is a kind of our workflow to be especially for fusion analysis in kits which will be launched later this year. If you look at some of the, let's say, technical corners of this kit, it has the ability to work with up to 384 universal dual indexes. It does include dual molecular barcodes. They are always present. You don't have to use them for analysis, but if you need them, you can use them. And something what is also a very important factor, especially for this kind of applications, either when you work with very low amount of input material, or with material which is somehow degraded or modified, that it does not or cannot be processed in a sufficient way, that we have that we have spent quite a lot of work on improving the conversion efficiency, meaning if you start with 1,000 molecules, that you have a very high percentage of molecules which are converted into functional molecules which can be used in the workflow. The other part, which was kind of in the mind during the development of the kit, was the possibility for automation. It does work on our 96-well platform on the Bravo, also on third-party 96-well platforms. But I think what is particularly interesting for low to mid-scale laboratories is this robotic solutions, which we have co-launched together with the XDHS2, with the XDHS kit and the XDH2 kit. This is the Magnus. The Magnus processes up to 80 samples, and it takes away all these steps of the library prep. So it's a very straightforward way of lowering the burden of training people and spending more or less a whole day on this process. Is you put in the sheared DNA, and the shearing will be brought on the system also later this year. But at this time, you bring in the sheared DNA, you put on the sheared DNA, you select the protocol, you hit the start button, and then after 8 hours, you can take out the ready processed library. It has a very clean layout because it's tailored for these two protocols, XDHS or XDHS2 protocols. Down here, we have the chemistry plate, which is cooled. Then we have the bait and buffer plate. And on the upper left corner, we've got the heat HSM unit for heat shaking and magnetics. So all these steps are performed there. And we have an integrated thermal cycler. So it's a really kind of robotic solution which works in a very nice and unintended fashion. The last segment I'm going to talk about is the panel part. When talking about the panel part, what is important to mention that we kind of in the background have completely overhauled the way how we design baits and also how we produce the baits. One thing is that we have introduced more sophisticated methods for designing the baits and placing the baits. One big, it is a buzzword, but it has been integrated. It is machine learning / KI to really pick the best performing baits, also with information from previous experiments. The other important aspect is that we also start to design probes against both strands. Previously, we have designed probes only against one strand. And overall, these improvements give a significant performance addition to the kit. So, for example, this is for a test panel of 3 MB. You are capable of reducing the number of probes required to cover a given design. You have a smaller design footprint, so you have less overlap. And probably the most important one, you increase the uniformity. And this one is not that significant, but for example, for the exome, we have a 30% improvement on this uniformity yield, meaning you have a much better read depth uniformity, and you need less reads to acquire certain read depths. And I just collected here the numbers. If you compare the latest exome release, the V8 against the V7, so we got a 20% reduction design size, 30% reduction of this fold 80 penalty, which yields to a 10% higher coverage with a given with a given amount of sequencing at 30x. If you look at the panels for the time reasons, I will keep it short. We have since a couple of years actually now, a panel which is comprehensive cancer panel, which is the oldest in that list. We have this set of Glasgow panels. This has been developed together with a collaboration partner at the University of Glasgow. These panels they have a different size, a slightly different, let's say, entity in mind. So the top one and the bottom one are for solid tumors. The HEM one obviously is for hemato-oncology. questions. They all have S&V possibilities, but also CNV fusions, MSI and TMB, which is obvious given the size they have. The other product group, which is kind of, let's say, product group which we constantly develop, is all-in-one. All-in-one because they also serve SNV, CNV, and translocations. We have a small one, which is just 200 kb, which is for lung in particular, but we also have one for solid tumors with nearly 100 genes. And we will launch end of this year a large panel with more than 500 genes, including a separate RNA fusion panel, which allows the targeting of of 86 genes for fusion analysis. And then last, but definitely not least, because this is still a big chunk of the designs we perform, these are the custom designs, which is one of the design types which is in use for this project. And I'm very happy to hand over, and thanks for your attention, and yeah, hand over to the next speaker. Thanks a lot.
Yeah. Thank you very much for this interesting kickoff of this webinar. We also already received the first question, so I would say, yeah, we can start with the first one. How much does the assay cost per sample, including the, no, sorry. Are the library kits for the pre and post capture application the same? Are they identical?
Yes, the library kits itself, they are identical between, you can use them either for pre and or post capture. You just have to take an eye on, since the pre-capture part are for 96 reactions, the post-capture part, because then the pooling kicks in, is for 12 reactions, so you have a kind of overlap if you want. On the bait side, you have to order specific baits, because specific bait sets, because they have different concentrations for pre or post-capture applications.
Okay, thank you. Then we will see you later then at the end at our Q&A session.
Okay, thank you. I'll switch off my camera, right?
Yes.
Okay, good.
Thank you. So, and then we will continue with our second talk and our second speaker, Dr. Van Liesefeld. So I will shortly introduce to you Dr. Van Liesefeld. He has a PhD in physics. From 2008 to 2013, he was working in a startup for ophthalmological medical devices and played a key role in the development of computer-guided laser surgery devices. And afterwards, he joined the clinical laboratory for genetic diagnostics, where he was responsible for establishing IT software development and bioinformatics department. This is also where he learned of the huge challenges, but also potential of NGS diagnostics, which finally led to the foundation of Limbus Medical Technologies. And since 2014, he's also the managing director of Limbus. So now today he will present us reconciling regulation and innovation with standardized software. So I will welcome Ben.
Thank you, Nadine. Well, probably the dominating topic in our discussions with customers has probably been the rising requirements in regulation that have to be applied to laboratory testing and genetic testing in particular.
I can't see you yet.
Yeah, exactly. So thank you for turning that on.
Sorry.
Yeah, so there's a slight delay.
Could you try moving a mouse? I think you're not in the presentation yet, so a little bit below. Sorry, No, I'm sorry, that was my fault.
No problem. So regulation has been a big topic. we are currently, as many of our viewers probably know, we're currently in a transition period from the old IVDD, the old directive, into the new IVDR. And there's kind of a deadline, a hard deadline in May 2022, next year, when IVDR will apply exclusively. IVDR brings some very important changes, particular to genetic testing. One is, for example, that devices meant for human genetic testing are now considered Class C devices. This is not a low classification, it's rather a medium or rather high risk classification. And this entails that the bringing to market of a Class C device, a notified body assessment is required. So a third party, a notified body, has to review the documentation of such a device. The regulation in Europe is also slightly different if you compare this to other markets like United States. While in the United States, laboratories are exempt from regulation, they are explicitly not exempt from regulation in Europe. Laboratories in Europe are required to use CE-IVD products wherever possible. And if laboratories want to deviate from that and want to use research use only devices in their processes, they must perform a process that's almost equivalent to a full CE approval. And I'd like to explain a little bit more detail what that means for laboratories. So if we're talking about laboratory developed tests, you're typically talking about the process, you know, a patient is being seen by a physician, the physician sends in the blood sample to the medical laboratory, and the laboratory returns a report after having performed its internal processes. IVDR now determines that once this medical laboratory uses research use only components in their process, the medical laboratory becomes kind of a medical device manufacturer itself and has to follow specific rules laid out in IVDR. And these rules are actually mentioned and determined in a very prominent place in Article 5, which defines what defines placing a medical device on the market or putting it into service. And Section 5 of this article states in general that the general safety and performance requirements apply. This is kind of a bummer or a catch-all phrase. The general safety and performance requirements are a long list of things that medical device manufacturers have to observe, and now laboratories have to do that as well. And in particular, IVDR determines that laboratories may not transfer those in-house developed tests to any other legal entity. They have to have a proper quality management system, and the proper quality management system in terms of medical device development is typically ISO 13485, also quite complex standard for quality management systems. And something that should not be neglected is there is a specific requirement that laboratories must perform post-market surveillance. So every laboratory performing tests must observe the performance of tests, regularly re-evaluate the performance, and document this process. And doing this from scratch without any assistance of any tools is probably very difficult and cumbersome. So if you're looking at your laboratory developed tests and each single component, you should look at how does the documentation, how does the level of compliance that the specific component provides help you in demonstrating compliance of your own laboratory developed tests. And there are different categories of products that IVDR knows. Basically, the lowest level of compliance is provided by research use only products. They basically provide no documentation at all that can be used for the IVDR compliance. Then there are general laboratory use components, which probably make life a little easier for the laboratory. IVDR then discriminates devices that are meant for performance studies only. These are medical devices that are kind of in a state before they are approved as a regular medical device. And then there are the regular IVD devices that provide the full documentation and that help laboratories most when demonstrating compliance of their own laboratory developed test. So, you know, determining which components to use will determine the effort and the extent to which you have to perform, for example, validation and verification and validation. Now, the varvis software, on top of being a CE labeled medical device software, also provides validation as a service. And this process works as follows. Our customers basically sequence reference samples according to our recommendations in our protocols. And those reference samples and the data from the reference samples are processed in the regular way with our software and uploaded into varvis. And we perform an automated validation process that exactly compares the output, the results obtained from varvis, to the published reference data known for the reference samples. That makes our software solution, or that provides basically a turnkey platform validation for our customers. And we discuss the many aspects of this validation service on our blog at blog.limusmedtech.com, and we will provide some references to those articles later on when we send out the webinar recording as well. So this validation covers not only the software, but the entire platform. And this is a performance test, which is compliant with the NGS guidelines from the European Society of Human Genetics, for example. But apart from this validation service, varvis also provides quality control data on a multitude of levels, which can be utilized for the so-called post-market surveillance, so for monitoring the quality, for monitoring the performance of laboratory developed tests. On a first level, varvis provides data with a focus on sequencing batches. So this is quality control across samples in a sequencing batch. The user can set specific thresholds and view at a glance how many samples are compliant or are deviating from those thresholds. It makes detecting issues in the wet lab very easy, and it allows to approve samples or reject samples from further processing downstream. Then there is case-specific quality control, and that means that when you open a case, you can get detailed information. In what way does your assay cover actually the transcripts that you would like to look at? So you can have information on that on a virtual panel level, on a gene level, or even on single exon level. And many of our viewers may know transcripts change all the time. So it's very important to make sure that for your specific case, that all the clinical targets are actually covered according to the current state of the art. And last but not least, in the specific case review, of course, varvis provides quality criteria for every single finding. And all this information is stored in a centralized database, and all the information is aggregated over time and can be monitored over time. So let me give you a few examples what the sequencing batch quality control looks like. One of the first things that you're probably looking at when performing targeted sequencing is the efficiency of target enrichment. Do you achieve the amount of coverage across all samples, across all bases in your assays, or is there potential to optimize? This is a cost factor in laboratories. Are you hitting are you achieving the right GC content with respect to with respect to the targets that you have in your panel? Deviating from the optimum here means that you're missing maybe missing important regions that you may have to fill in those missing regions later on. Also cost factor. You can break this down to single chromosomes. Here you can typically see issues with different input materials that you may face regularly. And varvis also provides an automated control or automated check of the sample sex that may be provided from external sources like a laboratory information system, or that is determined by different algorithms from the data, from the NGS data. And you can also determine any kind of contamination in your samples. So for example, we provide a very accurate way to determine the maternal contamination of prenatal DNA. So we were quite happy that when the University of Magdeburg approached us with a very exciting new application and asked us to become involved in the establishment of an assay for a very specific patient group delivering liquid biopsy data. And we were able to aid in the selection of suitable technology. We provided performance evaluation reports, and we assisted in optimizing the assay to achieve the best outcome, the best results for this specific clinical application. And with this, I'd like to hand over again back to Nadine. Thank you.
Yeah, thank you. Thank you for this very interesting talk and insights from software point of view. And I would say, let's jump right to the last presentation and our last speaker, Dr. Danny Schanze, and his talk, Matching Analytical Performance and Clinical Utility. So welcome, Danny. Hi. So I will shortly introduce you. Danny has a background in molecular medicine and a PhD in human genetics from the Friedrich Alexander University of Erlangen-Nuremberg. And since 2010, he is scientist at the Institute of Human Genetics at the University Hospital of Magdeburg, where he's also the head of array and NGS laboratory, and also successfully established NGS panel and exome analysis and clinical routine. And today, he will talk about, he will give us some insight in the clinical perspective and the evaluation and validation of this assay. So I will hand over the presentation to you. Just give me a second. And then, I hand over. Thank you.
Thank you, Nadine, for the kind introduction, and to welcome all to my talk at the end of the session for matching analytical performance and clinical utility. Only a brief overview on how we use NGS sequencing in our lab. So as most of the others, we do a lot of whole exome sequencing. So for studying neurodevelopmental disorders, intellectual disability, and many other diseases. But we also use targeted panel, and we use them for also copy number analysis. And we know that the coverage is much higher than, and the analysis for copy number is much stable. But today I want to talk about a strongly focused mutation, so the hotspot panel. So I don't know whether you can see there is a small dot within this blue circle. It's really oversized if we think about the KB, the size of the custom panel we talk about today compared to the whole exome. But there is a small dot, and you will see hopefully at the end of my talk why it's necessary to focus on such a small panel if we think of liquid biopsy and study other mosaicisms or really targeted somatic panels. So maybe I will have 2 or 3 slides, so giving an introduction to the background that is necessary for my talk today. So if we think on sequencing strategies, so we need different applications. If you look for a germline mutation, so where we use large panels or whole exome sequencing, we look for heterozygous or homozygous variants. So therefore, the limit of detection is above 20 or 30%, is much easier to detect these variants also on a raw coverage about 80 to 150x. So we only need data application of the pooled samples to separate the reads between these samples. But if we look for a solid tumor or a somatic variants, so the limit of detection will drop to 5 or 1%. So therefore, we need a different approach, so called UMI technology, as Andreas was introduced in the first talk. So the unique molecular identifiers and the raw coverage will increase dramatically. In the next slide, I will show you why this is the case. And also for liquid biopsy, maybe so the limit of detection further drops because the amount of DNA material we are interested in within the sample is quite low. So we have to focus on very small panels below 100 kb, maybe below 20 kb. I will show you later. And also the method that is needed for doing this analysis will change to duplex sequencing, and the raw coverage will further increase. So let's allow me to have a few words about the error rate. So we have to talk about that because we need the most accurate method to differentiate between the true positive variants and the false positive variants. And therefore the error rate is very important. So if you have a read, let's say 100 base pair. So if we talk about an error rate of 1%, then in average, one base in this 100 base pairs will be an error in sequencing or in amplification error that rises up. So therefore, the coverage is really necessary. So therefore, the change from the germline analysis to the somatic or to the liquid biopsy, the need for more coverage is very, very important. Because if you have statistical in each of these reads one error, then it rises up in different positions. But if they overlap in one position, and we have 100 reads, and in 2 of them the error occurs on the same position, then it looks like a variant of 2%. And decide whether this is a true positive or a false positive variant is very critical, especially if we look for variants in the liquid biopsy below 1%. So therefore, we need a higher accuracy in noise reduction and in controlling the error rate. And how can we do this? So let's say we have here a small barcoded molecule with a true positive variant. So on the forward read in green marked and in the reverse read in blue marked. So this is the true positive variant. If you do the amplification of this strand separated reads, there occur with, let's say, 1% error rate, there occurs some artifacts on different positions. If this occur later on in PCR amplification, there's really on different located on different positions. But if this occur in really early steps of the PCR amplification, maybe sometimes they occur on the same, because if it's incorporated in the early PCR steps, then they will be in all further reads or fragments that will be amplified from this. So now we can reduce only the forward strands and look for true positive variants here and the false positives and take them out. But in this case here, also the consensus strands for the reverse reads will rise up in this as a possible true positive variant. But if you combine these 2 strands, we will see that only this variant is a true positive, and therefore the high accuracy will be made. So if you took into account that we have in first here for our example and 1% error rate, therefore by using this separate consensus strands making, we can dramatically reduce this error rate and get at the end high accuracy reads with good differentiation between true positive and false positives. And we will see later on how this works. So at the beginning of our approaches or studies, we really ask ourselves the following questions that were very important. So is the single UMI technology sufficient to do this analysis, or do we need the duplex sequencing? What is the sensitivity if we think on the liquid biopsy, as I showed you in one of the first slides, that we want a level of detection of 0.5%. So what is the sensitivity? What's the precision of the assay? How much target region can we sequence? Can we do larger panels, or if you really have focused on a hotspot panel that is very small? And this is really important. And at the last, optimum compromises between clinical utility and the costs of all, yes, and the analytical performance. And also, therefore, I will show you some really exciting data that also addresses this. So what have we done? So we made 2 approaches. I call them approach A and B, and let's start with approach A. So therefore we use the single UMI, so-called unique molecular identifier technology from Agilent, also called the XTA HS1. So here on the next slide, you can see directly taken from the Agilent manual, an overview on how does the library that are generated for NGS sequencing looks like. So in the middle in blue, we have our target flanked by some adapters. And on one side, we have the molecule barcode that is 10 nucleotides long. So if you combine them, we can take over 1 million different unique molecules directly in the beginning and differentiate them in the analysis process. On the other side, we have the normal sample barcode for demultiplexing and pooling some samples within a sequencing run. As Ben said in his talk, we also need a reference sample. So we used a commercial available reference sample that was validated by different technologies, which contains 15 spiked in hotspot mutation variants with a variant allele frequency on 0.5 or 1% respectively. So we ordered them, we sequenced them, and later on we analyzed them and looked whether we could identify this variant, if what's the false positive rate, was it the true positive rate, and all the other parameters we will talk about later on. So we designed a custom panel, and we started with a very small panel, so nearly 37 KB in size that targets 144 region with hotspots mutation that we are interested in and sequence them on an Illumina NextSeq. And first of all, before we go into the detailed analysis, let's have a look on the usable reads. So first of all, we decided we looked into full data sets, which are part of the reads we can really use. And therefore, we have to think about the unique molecular identifier technology is necessary to differentiate between the true positive and the false positives. Because if we see that a variant arises or is tagged with two different unique molecular identifiers, that means that from the beginning, this variant was detected on two different molecules and therefore is more likely a true positive variant than compared to reads with variants that only tagged with one unique molecular identifier. And therefore, only half of all our raw data really fulfill this criteria and were therefore usable reads. That 50% of all the reads we cannot use for our analysis. So for some reason, we make a bioinformatician subsampling because we are also interested in how many samples per run we can analyze, and to not do this in several runs, we choose a bioinformatician way to subsample this data set to only 50% and therefore save sequencing costs. And we identified in this subsample set with only 50% of the reads equally distributed or for all our target region, that the useful amount of reads drops quite below 50%. So that nearly 40% we lose another 10% of the reads, and therefore it's an indication that maybe the doubling of samples per sequencing run will not very useful for our analysis. So if you have a look on the coverage, so how does our samples and sequencing reads are equally distributed and on which coverage? As you maybe remind one of my first slides with the coverage for the whole exome sequencing, we have an average from around 100x. In the somatic, we need more than 2,000, and also in the liquid biopsy, maybe more than 10,000 raw reads. So this is our processed reads, and we see that in the full data sets, more than 96% or 96% reach this 2000x, and in the full data sets, 83% the 5000x. But in the subsampling, so the data for the 2000x seems quite similar to the full data set, but here in the 5000x, so the coverage drops quite down to 60%, and also therefore indicate that this isn't enough for the analysis. Let's have a look on the detailed calling performance. So in this sample, as I showed you in the beginning, we have 15 variants that were spiked in at 1% or 0.5%. And if we look on the true positive variants, so these are the good news, we detect all of this really true positive spike in variance. So we have a sensitivity of 100% even for the 0.5 level, also for the 1 point level of variant allele frequency. But the bad news are we had quite a lot of false positives. So we have additional variants that are not in this reference sample, in that very accuracy and validated sample. So these are also false positives. So the precision of the assay isn't good enough. And therefore, it's clear that at this stage, we didn't move on because we need a better noise reduction and therefore maybe another technology to do this analysis. And therefore, we switched in approach B to the duplex sequencing. So compared to the first approach, we also have on this library XTSH2 from Agilent the target, but now it's flanked by 2 times molecular barcodes, so each side, therefore duplex sequencing, and they doesn't have 10 nucleotides, so they are 2 times 3. So in combination, we can take more than 4,000 molecules, and we have also unique sample index on both sides, and therefore we moved on with this. So we use the same reference samples, but by further reduction of target size, because we want to increase the raw data per sample, we only had 8 remaining of these mutations within our target regions, but at the same frequencies of 0.5 and 1%. And by reduction of target size, we only had 98 target regions with this hotspot mutation, and therefore overlapping with these 8 mutations in the reference sample, and the sequencing was done on the same Illumina NextSeq sequencer. So first of all, again, a look on the full data set and the usable reads, and by a reduction of the target size, so the custom channel was nearly half of the first one, we could increase the usable reads about 50, so it's nearly 58 or nearly 60%. So compared to approach A, we could slightly increase the usable reads, and that's a benefit for the analysis. But also have a look on the subsample data set. So again, bioinformatician subsampling of the reads to nearly 50% showed that we also comparable to approach A, we lose a lot of usable reads, that we have here only 40% compared to this nearly 60%. Therefore, indicating that maybe also in this approach, the increasing in samples per run will not work, but we will see. Also here, the comparison of the coverage. So we could slightly increase compared to approach A from 96 to 97% at the 2000x level, and from 83 to 85%. So the 2 approaches are nearly comparable. But if you have a look on the subsampling, so therefore the results are really dramatically drops down. So here in this of the 5000x level, we only have 33, so 1/3 of the sequences compared to 60% in approach A. But now have a more detailed look on the real data of the calling performance. So from the 8 true positive samples in this reference samples, we could all 8 true positive variants at a level of 0.5 and a level of 1% could be detectable. So the sensitivity of this assay and this approach will be 100%. And here, the further good news, we have no false positive, we have no false negative. So also the precision of this assay with these parameters, with a very low custom panel and a very high input read amount, is really good. So therefore, we got the best performance. Compared to the subsampling dataset with only 50% of the reads, so that was the simulation of doubling the samples per run, we see that also at the 1% level, we have the same results. But if we look on the 0.5 levels, we missed 1 true positive variance, what we are not interested in. And also we got 1 false positive. So this is a sequencing error that will be rise up as a true positive variance, but it isn't. So therefore, if we increase the sampling per run and have only 20 million reads as input for our analysis, then we must make compromises regarding the performance, what we are not interested in. So to summarize my talk, so we have to focus on really small target regions and using the duplex sequencing technology if we want to do the liquid biopsies. But also on this level, we have high sensitivity of 0.5 and a high precision regarding this large amount of input reads. And we are quite hopeful looking forward this approach of this really read consuming assay and workaround will be suitable to study some liquid biopsies and some of the diseases we are interested in. And so far I presented a really newest data. We are looking to make some custom panels, so we have to decide not to make one larger panel, so we have to decide to make several small panels to reach this high input for data analysis. And therefore, we are looking forward to maybe present in one of the next webinars our newest information on further developing these tests on liquid biopsy samplings. So thanks a lot.
Okay, thank you very much. Thank you for presenting your work and sharing your data with us. So we already received some questions. Just as a reminder, if you have any questions, then please feel free to enter them in the chat. And we have all our speakers here to answer them. So Andreas is just missing. So maybe if you want to, also you can turn on your camera. But let me, hi, there you are, all of you. So, okay, then first questions. We have two questions about the pricing. So one is, I think this is for Danny. How much does the assay cost per sample, including the software?
Yeah, it really depends on the size of the custom panel. So also maybe this question is to Andreas. So we started with low kits with the only 16 reactions to make all these validation steps. And after validations, we decided to go for the larger kits with 96 samples that also have a positive effect on the costs per sample. So directly, I can't say it. I have to recalculate it because in this step, we are not focusing on the money. Yeah, it's nice to say this, but first of all, we were interested in what is the most precise and most sensitive method to do this analysis. And afterwards, after defining all the conditions, the parameter for sequencing, the parameter for the custom panels, we want to decide how we move on. I didn't say it, but we also for money reason or cost reason, we decided to go for a mid output on the Illumina next SEC. So therefore the cost reduces dramatically compared to the high output, and also for further smaller turnaround time. So on a high output on the Illumina sequencer, we can sequence much more samples that maybe also end up in a lower cost per samples, but always collecting samples and wait 2 weeks to have enough samples for a large one is not the most accurate way in thinking of diagnostics. So far we are in a research use only step, but we plan to move on and do this as a diagnostic approach in further. Also with the help of Limbus and regardings with IVDR next year. And also therefore we were very thankful that they helped us to validate this assay in a really early step, and that we don't want to do this in May next year.
Okay, thank you. I think this is also for you. For now, you have not run liquid biopsy samples and generate data to present. And I don't know if this is also regarding this, pre-capture pooling or post-capture pooling effects tested and seen for the precision?
So far, we don't have tested pre and post capture. So we doing only post capture because so far we tested ideal materials, so all of the controls. In the next steps, beside tissue material, FFPE material, and further CF material, we also want to see how does this performs, how does influence the quality of the material. So these are the next steps plans, and we already sequenced one run, but the results are outstanding. So I could not present this data today. So it's maybe is a first talk in a series of 2 talks, maybe 3, I don't know. We have to discuss about this, but this will be the next steps. So far, we have an optimized material, we have an optimized workflow. We are believed that it's with a higher sensitivity, with a higher precision, and this is the start point. So now we can further look for the influence of other parameters, of amount of DNA, of quality of DNA, of the integrity of the DNA, regarding really high molecular tissue samples or maybe more degraded FFPE samples. So we believe that not only for the liquid biopsy, so for the CF DNA, this approach will be most suitable, but all for the other materials. And this will be the next steps where we plan to do several sequencing runs with different materials and also the quality of the materials. But therefore, I have to postpone these results to maybe another talk.
We would welcome this. Follow-up question, are there varvis-generated clinical data to present?
From liquid biopsy, I mean?
I think this is yeah, regarding the.
I mean, we don't generate the data ourselves, right? So it's always a partnership with our customers. I mean, we are planning to illuminate more the clinical aspects, the clinical application of what you actually then interpret with the software. So this talk was rather very technical on the analytical on the analytical side. We will have another talk on at ESHG probably about the clinical application of these assays, and then you can probably see better the software in action.
Okay, thank you. So then another pricing question. How much does the validation service cost?
So this probably the validation service of varvis is free of charge for our customers. So so if you are an interested user and would like to evaluate our software, some of our customers have performed the entire validation process during these 2 weeks of software evaluation that is provided free of charge. But anyhow, even when you're a long-standing customer of ours, when you're introducing a new assay, we assist you with the validation and provide the performance evaluation for free.
Okay. And another one, I would like to know more about varvis for NGS data analysis. Can you provide an overview of the pricing for validation of DNA and RNA targets using the focus panel?
I'm not familiar with the focus panel. Is this in a? So I'm just so if it's just a general term for a focused targeted panel, the costs for the routine diagnostic for processing the data depends on the volume. So I can't give you a general answer here. Just as a rough benchmark, it makes sense to use varvis from a volume of 200 samples per year. And kind of the price per sample drops the higher you can, or to a higher number, the higher the number of samples is that you can commit to per year, because we also purchase resources in advance, and we pass on those cost savings. Yeah, so validation, again, as I said earlier, is free of charge.
And we have a question for this, validation costs for non-existing customers.
Well, I mean, so the validation, of course, or an essential part of that validation is our software. So the performance evaluation that you obtain is for the entire platform, consisting of the wet lab and the software. So the results may not be very meaningful for somebody who's not using our software in routine diagnostics.
Okay, a question for Andreas. What are the minimum input quantities for the XTHS2 workflow?
So according to our protocol, the, let's say, range is 10 to 200 nanogram, with a sweet spot probably around 50 to 100. But especially when it comes to amount limited material, we do see people which are driving it significantly lower, even with questionable material like FFPE. Of course, what people should have in mind is if you really go down, you have a limit in your number of molecules or genomes which you add to the reaction. So you may bump into some issues, some statistical considerations which you may make upfront, which cannot be fulfilled if you just have, let's say, 500 available molecules which can be processed, because then you bump into that limit kind of naturally.
Okay. And
I have a question.
Yeah.
Can I ask? It's to Danny and Ben as well. When it comes to liquid biopsy in tumors, I think you have, say, two sensitivity aspects. One is that you have to measure against the background. Yeah, so you may, your tumor cell content might be just 1%. And do you have to apply mentally another layer where you say in solid tumor, you talk about tumor cell content? Yeah, because the tumor, or is it just tumor cells which are indeed tumor cells, or is it, let's say, a subpopulation, for example? Is 1% or 0.5% enough to detect rare variants, given that your material is by definition diluted? Kind of a general question regarding liquid biopsy, probably.
Yeah, it goes in the same direction. So we also started with the germline. So the germline is a normal analysis we do, but also we face the problems regarding the somatic analysis. We are not a pathologist, we are human geneticists, but also we have diseases where we look for more mosaicism. So it's a kind of a somatic analysis, because also a small proportion of the cells, and we not always can pick up a biopsy out of the brain or some positions within the body where we are not really easy can address and took a biopsy. So therefore we face the same problems. So if you believe on a tumor, if you have a high tumor content of 80% or something like that, then a 20% variant or limit of detection is easily seen that you have nearly 15 to 20% variant or allele frequency. So if it drops down to 50%, it will be diluted by twice. So we only see the 7%. If we drops down further the tumor content to only 20% or something like that, and it's nice to have this amounts from the pathologists. We not always have them. So we also face this variant frequency of between 1 and 5%. It's the same for the liquid biopsy. In liquid biopsy, it's not the content of the cells, but the rarity of the molecules itself. Because the small tumor within the body somewhere will only took some of his DNA to the fluids in the body, in the blood, or in other liquids. So therefore the content itself is really low, and therefore the variance we are looking is diluted by background. So in CFC DNA extraction, we focus on the plasma so that we get rid of all the cellular fragments that maybe can make the main background. We focus on small fragments, so we have some size selection and extraction, that we more and more enrich these small fragments that directly comes from the tumor and is represented the cell-free DNA fraction. So therefore we try to enrich on this and therefore increase this amount. But we know that also at the end, we must face that we have only 1 to 3% of variant allele frequency. We have not only sequenced so far these control DNAs, we have also a few real-life samples with known mutations that were identified in prior analysis using also NGS-based technologies, and we compare them to the cfDNA, and we could validate this analysis, but this is really preliminary and have to be proven in the next runs. So therefore, I don't want to say much about this, but we can validate this at a level of around 1 to 3 or 4%. So in the CF samples, we did not analyze or see a variant allele frequency of 10 or 15% compared to some somatic samples. So it's quite below. And we also see some targetable variants or somatic variants where we really can make a therapy for the patients with only 1%. But without knowing this variant, we cannot offer any therapy to the patients. So therefore, it's a large benefit to do this analysis on the liquid biopsy, and therefore the identification of some variant that are targetable. So this is the main approach. So the patient and the outcome for the patient, so that we can identify the disease causing variant also in this small amounts and at this level of detection.
So I can add to that. So the size selection is would also be something that from the software side that can be applied. Of course, it's more cost efficient to do the size selection before sequencing. You could also do size selection of fragments after sequencing, but then again, you would filter out data that you sequence before at high costs. So it makes sense to do the size selection in the beginning. But kind of the size, that's kind of one of the leverages that you have to enrich for those variants that you're looking for.
Okay. Interesting to think about filtering it out on the software level. Yeah.
Well, we've done that, and there are publications about that actually. But yeah, again, you are throwing away data that you generated under pain. No, not under pain, but as a bioinformatician, it hurts a little bit to disregard data, right, that you have already obtained. But it's not. But you cannot make use of it, and you will only regard those of a specific size.
Also from our side, so in the beginning, if we see the first results and we lose 50% of all the reads that are not usable, okay.
I feel you.
We increase this amount of usable reads by changing the technology, so we are quite happy. But we need a lot of input reads. So as I showed you, 20 millions are not enough. And if you compare this to the capacity of a NextSeq or a NovaSeq, so therefore you can calculate how many samples. So that are not hundreds of samples. You can sequence also with this very, very small focused custom panel.
And what is the suggested?
It's necessary.
What is the suggested read length, which works best in your hands? Is it 2 times 100 or 2 times 75 or 2 times 150?
We use 2 times 150. We always goes for the larger sequencing lengths. It's, to our opinion, has some advantage in alignment and also in the cost efficiency. So therefore, so far the probes we have tested, they allow us to sequence 2 times 150. If we go for further degraded FFPE material, we will analyze this in the next steps. How does influence the sequence ranks and maybe also the quality of the sequence reads.
We see across very different types of assays, we see a benefit of going to longer read lengths, even if the library that was prepared has an average length of only 140, 120 nucleotides. It's of benefit to use the longer reads. Just looking at analytical performance, how well are you able to determine, in particular in indels, right? More complex variations can more easily be detected using longer reads.
Sure. Yeah. Okay, yeah. Indels is a good point. On the other hand, I was a bit surprised to hear that, especially when working on by definition small DNA, that because you have a lot of, if your average read fragment length is 150 or insert length is 150 or below, You have a lot of overlap reads, which in most of the pipelines get anyhow kicked out or just seen,
But
so
I would say modern algorithms take that into account, so they are
okay.
able to deal with that and utilize the information.
Okay, cool.
It's also useful for the consensus making. So if they overlap bidirectionally for all the 150 base pairs is much better than if they overlap only for 50 or 70%, because making the consensus strand specific and then over strands. So we need this overlap to really differentiate between the two positive and the false positive variants.
Okay, nice panel. Not much to do for me. Another question, an interesting question for Ben. Because of cloud-based services, which cloud server service you offer? Any restrictions you see in Germany for data safety with data outside of the lab?
So I can speak from my experience as having been responsible for data safety in a laboratory, in a clinical laboratory before. The task for us as a dedicated service provider for a specific application, the task for us to provide security is actually easier than for a clinical laboratory. In any corporation where you have a very diverse infrastructure with many different services, different applications, and people working in a laboratory with computers know that probably. For us, it's much easier because we only have this one, the single service, and we can monitor and secure the service very nicely. We are, of course, GDPR compliant. We have demonstrated to be compliant with, in audits with many of our customers. So I would say 50% of our customers are German university clinics, and they are known to be very rigid, or let's say very, very diligent in their assessment of data security.
Okay, last but not least, so one question open. I think it's a good one for the last question. Do you think that this assay is suitable to be performed in routine diagnostics for Danny?
So at this stage, I think it's the best start point. So we have proven that we have a sensitivity of 100%. We have proven that we have a specificity. The precision is at 100% for these tested and known mutations, so that we can now move on in testing the different materials, as I showed before, or I said before, that we can prove for the tissue material, for the FFPE and the CF material. So this, from the starting point, it's the ideal starting position. We will see whether this assay really works on the different materials, and after testing this, then I can say, yes, it's ready to use in a routine diagnostic, or no, we have to make further improvements or something like that. But to our point, it was very, very important to start with the technical performance before we go into the materials and so on. We never know what materials will be sent from the clinicians. We always say, yeah, don't send us the FFPE materials. We want to have an cfDNA, or we want to have a fresh frozen sample or a tissue sample. But sometimes it's not possible. So therefore, we started at this point. And yes, I hope that this will become a routine diagnostic test in our lab.
If I continue from the how to say, the panel and library manufacturer perspective, we are of course also having this in mind, and positioning ourselves and getting the corresponding IVDR or CE-IVD labels on the kit and the panel side. I would say by the end of the day, to say, does it have a chance to get, let's say, full compliance or an IVDR Class C label? I think one has to put together the entire package, including analysis, and run a rather large cohort of samples, of real clinical samples, and to finally come around with the sensitivity and specificity of that assay. Let's say with a given set of settings, because right now, due to the let's say to the benefits of the software which are in use, you can adopt a lot of things, and since it also is, I understand, somehow work under progress. But when it has to become a clinical validated, or let's say a clinical ready platform with a proper label, one has to lock the parameters, but I believe, let's say, with all the tools you have for QCing, you can reliably tell whether it makes sense to trust the data which you have used as an input, and then use your pipeline and then validate the whole thing, and then we'll see how good it performs against, in terms of these, let's say, parameter sensitivity and specificity against other technologies on the market.
But this is certainly an assay that has been in the mind of the regulators when they specified the rules for laboratory developed tests. It's an assay for a specific patient group, and those patients should be served, right? They need their specific diagnostic tools, and I think that's the right approach that University of Magdeburg takes, and pushing forward the diagnostic possibilities here.
Yeah, definitely. Great.
Also, there is no commercial test or a custom panel or a commercial panel available on the market to really rule out this patient group. So therefore, we need a laboratory developed test that is really focused. And as I presented today, it's not suitable to do a whole exome sequencing on this patient group or on this liquid biopsy. So therefore, we have to focus on really small panels. And yeah, sure, if we change the content of the panel, we have to begin at the beginning, so we have to make all this validation again, but we have this commercial available reference samples, and if we rule out the whole process once, we can repeat them for the second panel and for the third panel, and we will do this in future then for the next ones. But this is really the beginning, also the beginning of the documentation of the validation, and also therefore, and if we don't have this precision and sensitivity, we don't move on. We can move on, because if this not fits, then all the other parameters this influence the outcome of the test regarding the sample amount, sample quality, and all the others. It doesn't matter if the sensitivity and precision does not fit.
Yeah, but then your validation thing really kicks in. I think that's pretty cool. Yeah, great.
There's something left to do for the next month.
And I'm sorry to say that, but there was still a question coming in, 2 left. Is there any possibility that you will make a panel for homologous recombination deficiency? Sorry when I pronounce it wrong.
That's a question for then, Denny. Homologous recombination deficiency application for you?
I don't know exactly. So we are not focusing on this. So we are interested in on really small nucleotide variants and small indels in our research, because so far for our patient group we are interested in, so this doesn't take into account. So I don't know exactly. I didn't think about that so far. So maybe you, Andreas, have some ideas from your side of the manufacturer.
We have projects working on that topic, and but since it is these are custom projects, I'm happy if you're interested, we can try to make the link between the customers who are doing that and see what if they agree. But this is definitely something you can address with a platform. The interesting parameter, I don't know if this was the intention of the question, when it comes to, because the current approaches, especially when you talk about HRD, this is based on looking at, by the end of the day, at LOH events across the genome, which require a certain footprint of the design. If this has to be combined with liquid biopsy, given what you have just nicely explained, if this is continues to be an attractive approach, kind of, let's say, neutrally said, I think this would have to be validated how much you can reduce that SNP backbone, that it still produces reliable results for LOH detection, which sensitivity, which size range is, you have to maintain. This has to be seen. But so far, this is done on FFPE material, and there we have excellent results, I can tell. Yeah, but on liquid, not yet.
Yeah, but sequencing a large background in context of liquid biopsy would dramatically enlarge the panel size.
Exactly.
So that's not that what we are interested in, to keep it as small as necessary to reach this high input amount and get an acceptable cost efficiency per run or cost per sample.
So long story short, for
what
solid,
we are
yes,
most.
for liquid, we need a big sequencer.
We will see.
Last question. Do you think that, I think that is related to the question before and your discussion before. Do you think that continuing with these this best knowledge panels will work in the near future by tuning them for best diagnostics possible under regulation?
It's hard to say. So focusing on this really small panel is the best way we can offer the best analysis at this time point. We will see how this will work regarding the IVDR regulation, whether we really can make this a laboratory developed test. We are not interested in to do this for an CE certification or something like that. So this will always be a laboratory developed test, but we will see. So we started this project because we have the need for this analysis for our patient group. And also we want to establish a collaboration between the clinicians and the analysis in our laboratory, so that we are really in contact with the clinicians with the new coming up mutations that also 2 weeks ago there came up a new recurrent mutation in a new gene that have to be incorporated in a new panel version in the future. But so far we are moving forward on this step and the validation with the next ones, because we could not change the setup of this panel each week or each month. So we're looking forward for an re-evaluation each year or each half year. We will see, because this field we are interested in is strongly moving forward and is enlarged with coming up new mutations. So also we are focusing on this. We will see how this will change in future. I cannot say how this moves on and whether it's overruled by all the regulatory needs from other sides, or maybe from a commercial available test from any of the companies that then maybe is IVDR certificate and really fits the need for the analysis for our patient group. We will see. I cannot state on this yet.
Okay, thank you. So there are no questions left. Then, I would like to finish. So thank you very much for this interesting talk, for this interesting discussion, for presenting your work. For more information, please visit varvis.com, or as Ben already mentioned, we also have a blog. I added the links to the chat, and we will also send it to you in the follow-up mailing with the recording. And if there are still some questions left, if you're watching the recording, forward it, and there are questions, then please feel free to contact us. If there are questions for the speakers, then we will forward them. And then, thank you very much. Nothing left to say. Thank you for the speakers. Thank you for the audience for the interest and questions. And
thank you. Thank you, Nadine.
Have a good time. Thank you. Bye.
Bye-bye.
Bye-bye.
Bye.
Bye.